Hello, everyone, and welcome to today's live broadcast, Beneficial Microbes, Sharing of the Microbial Informational Library, presented by Dr. John Thomas, Professor Emeritus, West Virginia University School of Medicine. We are excited to bring you this educational webcast presented by LabRoots, the leading scientific social networking website provider of virtual events and webinars advancing scientific collaboration and learning. I'm Julie Simmeroth of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located in the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This is an educational webinar and thus offers free continuing education credit. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credit. Please join me in welcoming Dr. John Thomas. I will now turn the presentation over to him.
Well, hello, everyone. This is John Thomas, and it's a pleasure to talk to you today about a topic which I've spent a long time studying and evaluating, and I want to hopefully educate you to the whole world of microbiology and how much it's changing who we are and what we think about ourselves. I'm going to do this with about 5 sections, but right now I just want to make sure you know that I've also got some slides in here of me. I'm a skier and a national ski patroller, and one of the points is that although we're going to cover a lot of information, it's still a very important point to enjoy what you do and love most. I've been a professor for a variety of times at a variety of places, but I want you to realize that it's important that I've been able to integrate 3 very unique but integratable thoughts. One is I've been in medicine, been in dentistry, and a clinical microbiologist, which gives me, I think, a unique perspective about how what we're going to present today is so influential and so important. The first part of my introduction will really define how the world is changing and who we are in it. And what I want you to realize is that there is a theory which has evolved in the last 10 to 12 years, which describes co-evolution of microbes and microbes with man. And the reality is, I came up with the word, I've heard it before, that we are involved in a dual citizenship. The reality is that you and I are both parts of the genomic total, and that there's a macro part and there's a micro part. And the new concepts of dual citizenship really address this generation as being a holobiont. And we're going to talk today a lot about the microbes being beneficial that are the micro part of the holobiont, but it is the macro together, and there are shared benefits, and that's very important. There's a lot of talk about this, and the references here I think will give you some clues, but we need to move on because I want you to recognize that in my theme, it's also been one that was addressed by a very famous speaker who came across the idea that we must reassess our self-concept, who we are, because now it's very recognizable that microbes matter. And the reality is that when we together form a combination, one half of us essentially genetically is going to be who we think we have been, but the second part will be who are the microbes and what genetic strength they provide. It is a combination of both. If one looks at the greater part and for a greater definition of this who we are and the hologenome being made up of the macro and the micro, one can take a look at this picture and sort of describe who the components are. But it's important to recognize there are already 3 parts then that make us who we are, and that's the person who is the Homo sapiens. It's the microbiome, but I will emphasize that we have the mycobiome, or the fungal part of our holobiont world. And then we have the environmental. That is who we are around us. And those three things together will help us create an environment that will move forward, recognizing what I try to put here, and that is we are really libraries of information. And the holobiont is a combination of libraries that are human and libraries that are microbiota addressed. And the reality is, again, if we look at features distinguishing each, the human is about 10 to the 12 eukaryotic cells. There are at least 10 to the 14 prokaryotic cells. And if one looks at the gene pool that these represent, the library of information, essentially humans have about 23,000 genes, and the mycobiota, that is bacteria and fungi, have at least 10 million genes. And the reality is that if we examine ourselves in a genetic sense, the informational potential of that microbiome is quite remarkable and needs to be further evaluated. Which then drives another question. We, as you look at the bottom of the slide, are essentially described by, again, the microbiome of about 10,000 different species of microbes. To begin to focus on how important our microbiota is and the mycobiota, the bacteria and fungi, we need to begin to put in a reasonable system, and we've sort of gone back to all of us looking at our taxonomy. And the bottom line is, if you look at phylum and class and perhaps family, we get a much better idea of how we can measure changes that can be reflective of disease and or health. And there are really 6 of the phyla that we have found, and I will probably describe many times throughout this paper. But the bacteria that are listed in front of you as phyla represent truly profilable microbes or organisms that will give us some clue as to a change from what should be or what has been normal in our age, from the development of our microbiota at birth to the development of the microbiota as we age. In this diagram, we clearly see how unique each of these 6 phyla, described in a zone throughout the human body, changes and is remarkably characteristic of or associated with a particular region. And these regions are blending. And I remember, as I was taught, we used to think of ourselves as anatomic locations. And now I want you to change that a bit as we begin to address ourself and to recognize that we flow from one site to another, and the microbes that inhabit that, defined by phyla, are part of that. So if we think about ourselves and our microbiota in the microholobiont, who are these beneficial microbes, and how are they able to do for us what is in essence a shared responsibility? And what you'll see here is a definition that I've been working with, and it is that it provides the beneficial microbes, often associated as probiotics, but let's be a little broader in sense. They provide a benefit to hosts in a meaningful way, recognizing there is a shared benefit. And I think that's incredibly important. Not only does the microbiome provide information to us, we provide information to the microbiome, the dual citizenship. And the reality is I've come up with intelligent symbiosis would be a meaningful way. And listed here on this slide are 7 characteristics that are a result and that are an interface between our dual citizenship of microbes and human cells. There are quite a few, as one can imagine, beneficial microbes. And although this deals with medicine and dentistry, and please notice that I add the word dentistry, it's important to recognize that beneficial microbes are apparent in other parts of our world, particularly in soil, in plants, and in industry. But in dealing with medicine, I've classified them in a large way of those that are therapeutic, those that are predatory, those that are dealing with signaling. And there are 2 examples shown on this slide which help again begin to organize our thoughts that in fact a bifidobacterium has been used as a search and find marker for a particular kind of bladder tumor. On the same hand, a particular kind of cancer has been dealt with in a detection and predatory manner by essentially detecting a bladder tumor also, and by addressing its demise by essentially being able to replicate, and in so doing, that is, the salmonella begins to destroy the tumor itself. So beneficial microbes can be a variety of things with a variety of purposes. One of the most common is lactobacillus. The other one is probably bifidobacterium. And if one looks at what things they possess and how they work, one can begin to define some of the features that would highlight a good beneficial microbe and a bad beneficial microbe, or one that is not so capable of being beneficial to the host over a period of time. It is these features that help us begin to distinguish who and what microbes might be the best as far as beneficial bacteria, and particularly beneficial fungi. However, I've been in science a long time, and I've been probably half of my life has been dedicated to the study of biofilms. And one of the things that's now becoming apparent is that a good beneficial microbe, a good probiotic, is one that forms a very healthy biofilm. The photograph in the upper right-hand corner is that of an endotrach and a biofilm within the endotrach, and the four areas on the lower right-hand side describe how we have looked at and defined a biofilm as in stages. And the reality, though, is that a biofilm is, as outlined, a developmental biology. It's an immobilized enzyme system, and it's steady states. But it's a community of microbes, generally mixed, that are able to perform tasks that greater than the individual, because there are components that help reinforce each other's part to the system. Before I go to the next one, I do want to emphasize, though, that as we talk about the microbiota and we talk about the mycobiota, it's very important to realize that we are as much colonized with fungi as we are with bacteria. And the reality is we know very little about that. And I'll discuss this a little bit later. But in the original studies of the human genome and the bacteria associated with those regions, we sort of left out the fungi. And that's an important feature that down the road we will need to address as we get a more complete picture of who we are, both from the human and from the microbial population. Well, if microbes are so important in fungi, how do they work, and what is this shared option that we now have? Benefits to the bugs and benefits to the human. Benefits to us and who we are, but then who we choose to share our cellular mechanisms with. What is the benefit of that? And in our original studies, as this has evolved, we began to think of ourselves as the beneficial microbes in three distinct areas, although, as I pointed out earlier, we really want to make sure you realize that this is a region that it fuses into other regions. But the reason these slides are to the right, oral and gut, or gastrointestinal, are clearly the regions that we have now the most information about beneficial microbes, and places that, in fact, we might be able to use the intelligence of microbes to help us become better in our dealing with health and abnormal consequences. Obviously, as you've all heard, the majority of the microbes that we have initially studied have been in the gastrointestinal tract. And that is partly the reason that we begin to understand how there is a response and how there is an attitude of microbes being able to communicate with other distal parts of the human body. And it's still being addressed. This is not something that's clearly been finalized, but it's something that we're getting a handle on, and we're getting clearly an opinion that this is quite a complicated system. And the reality of axial communications between microbes and Homo sapiens cells has been, at least for the moment, divided into four components. In the upper left of this slide, we have the capacity of microbes to direct signal. On the B, we have metabolic signals are coming from the host, which help direct the microbes to enhance or reduce a particular signal. C, species-specific signals come from the biofilm imagined cooperation, and that the bugs within and the microbes within the biofilm interact as a community and alter the axial signal coming from cells in response to human cell signals. And then the last is D, the axial conditions, where there is a direct bacterial species which elicits transcriptional responses. In other words, when humans and bacteria are together and sharing benefits, The transcription of DNA to RNA to protein is coordinated so that there is a shared benefit to both of the members who are in this pathway. So what are the mechanisms? Knowing that there are essentially at the moment 4 pathways, what about the mechanisms of action, and how do beneficial microbes, particularly probiotics, of which we know the most at the moment as representing mechanisms of action? You can see here listed at least 5. And one of them is very obvious. They can inhibit growth of pathogenic enteric bacteria. They can block attachment, particularly in the GI cells. They can downregulate certain toxins being produced by pathogenic microbes. They can improve the epithelial mucosal barrier function, particularly in short-chain fatty acids in the gut. And really where most of the current research is going, they can alter host immune response. They can either upregulate or downregulate immunologic capabilities. And that is where there is at the moment such a huge focus, given that although the beneficial microbes and potential use of probiotics are important, that is where there seems to be a clear direction to get the best of our dual citizenship and of the dual capacity of a shared microbial world with our human cells. The last part of this section then is really how do these cells, having that we now have some mechanisms, we now have an idea how they actually communicate through the systems with potentially 4 routes. One of the growing recognitions is that at the end of this communication scheme needs to be an actual action group of molecules. And it seems to fit very nicely to what has been described as a yin-yang hypothesis, or good and bad, hot and cold. And the bottom line again is there are those consequences of this cascade which will be beneficial to the host, as shown in the bottom in the light blue color, and that those that are absolutely deleterious to the host. So in, again, the simple sense of how beneficial microbes may be beneficial, and how if the wrong microbes are present and the shared gene expression is not beneficial, it can be essentially deleterious. We now have what we're going to call the yin-yang hypothesis, which has been first so well based in cancer and cancer chemotherapy, and now coming over to the Hologneomic theory and the concept of dual citizenships. Well, now we really have the opportunity of addressing that part which I find most engrossing, I suppose, because it helped me think my thoughts into a hopefully organized format. And I'm going to talk about something that I had thought about, but it's going to be called the Microbial Clock. And the Microbial Clock, as I'll mention in a moment, is key in sort of recognizing that if in fact there is this dual citizenship, good things and bad things can happen over time in sort of a cascade of information. So I'm going to talk about the clock. I'm going to talk about one infectious disease, and then we're going to talk about the areas of our research most recently, recognizing again the beneficial microbes, and that is wounds, and most recently aging. It became apparent to me that as we began to synthesize all of these separate entities in human disease and look at them particularly in a manner of endogenous diseases, you could begin to look at it as a clock. And I decided the clock could go in essentially 4 areas defined by colors of green, yellow, orange, and red. And those clocks would help me begin to place into the development of Homo sapiens and its corresponding microbiota diseases that are associated with age and how these impact particular time frames in our growth, and recognizing that there's always been this disparity of how these might be linked. What started me on my path to putting this together was Dr. Blazer, who said that connecting multiple diseases through a single framework or underlying cause was a perhaps more palatable way than trying to define each one separately. And I list, as you can see here, all of the diseases that are 10 of the diseases which are human, but follow a path. And again, that was very interesting because if you take those diseases and look at this microbial clock, but now begin to emphasize the disease and the associated changes within the microbiota, and look at again those 6 microbes or phyla that represent a cluster of microbes or groups of bacteria, one can begin to see that through time there are changes, as one would expect. But the reality is now we begin to have a target that we could either be associated with a marker, a biomarker, or one that could be associated with a deplumation or a decrease of the microbes, and from that we could begin to think about adding bacteria back into our organ systems, given that there is this potential of now being able to profile good bugs and bad organisms, or good phyla and bad phyla. There's a really interesting, hopefully, diagram that will make you think a minute, but taking this approach of our microbiota and the concept of a healthy and then a deleterious microbiota or dysbiosis in the gut. We have on the left-hand side sort of a hamburger, and we have the consequences of the problems that can occur in eating that, and then the kinds of GI microbes which might be enhanced. On the top, sort of those that are beneficial, on the bottom, those that are not. We can have then the consequences of the mechanisms we talked about, and then ultimately the target tissue inflammation, again, yin and yang, as the consequence of what we have done to the microbiota of our gastrointestinal tract. You are not what you eat, you are what your bugs eat, is perhaps the sort of loose-ended but sort of fun way of explaining what we can look at. And one of the major tracks, and unfortunately particularly here in West Virginia, is the recognition that obesity is our number one problem in the age group of what I have called the orange, growing through the adolescence. And if one addresses obesity and one addresses microbiota and the phyla, one can see that weight and diet and microbes, particularly the phyla of Firmicutes and the Bacteroidetes, are those that are associated with those that have a obese figure, whereas those that are having less of a obesity and not the problems associated with weight loss or weight gain have another kind of phyla-enhanced environment. And the reality is that phyla-transmitted or microbes or gut-associated microbes can transmit this situation, which begins to point out that microbes are the players in these diseases. As a clinical microbiologist, however, I really wanted to deal with 2 very appropriate diseases that I am very interested in. And one is wounds, chronic wounds, wounds that are associated with the breakdown of tissue. And it began to be apparent to me that this process was, in dealing with our colleagues, both a disruption and a reconstruction. And the disruption could be handled by doing something different to disrupt the pathogenic biofilm, but perhaps reconstruct a beneficial biofilm in these stages that are represented here. It came to me that this was important because in one of our studies, looking at gauzes and looking at particular anti-infectives impregnated into the gauze, is that if you see in the upper left-hand side, we have actual gauzes that are impregnated with a particular compound. When we tease those actually apart, although they were releasing the compound as shown by the clear zone, on the right-hand side you can see that in fact there is a growth of the biofilm material from the inhibitory impregnated gauzes, and that there were biofilms within the dressings. So that in the middle of the diagram you can kind of get an idea that the wound bed has a biotic, a biofilm environment, and the dressing maintains a biofilm environment. And we clearly thought that if we could stop this ping-pong effect, we could address a better enhanced healing process, which led us to go to looking at different and alternative sites. And so we basically came up with a comprehensive wound care should in the destruction part deal with the classic debridement suctioning, maintenance of the pH, and then a disruption with some form of anti-biofilm product, but then in the middle addressing reconstruction with that particular group of beneficial microbes or particularly a group of selected probiotics. And what we began to think about was, could we make a better gauze, and could it enhance the beneficial microbes which were a part of the original tissue to begin with? And we began to think about having a smart gauze, and if you look in the lower left-hand corner, you'll see that the smart was for a symbiotic, both pre- and probiotics, with mixed species that could produce a biofilm, that were addressing antibiotic resistance by not inducing that resistance, and by having a therapeutic response particularly associated with antibiotic stewardship. We began to look at microbes, particularly lactobacillus, and all I want you to recognize is that if we use single microbes growing on a wound bed, multiple microbes of 2 microbes, or 3, and then use Lactobacillus rehteri, we got a varied response depending on how many microbes were in the wound bed, which would be logical. We focused then essentially on what was our next gauze, or smarter number 2. We looked at biofilms, and we used basically a live dead stain, and if you look at the right-hand side, you'll see the consequence of a Lactobacillus rehteri probiotic in a wound-encased gauze, and you can see that over time we could reduce not only the average numbers but the biomass, and whereas green is living and red is dying, the concept of putting on a gauze, addressing a wound bed with the inoculum being a probiotic as a beneficial microbe would not only begin to have some stimulatory capacity, but it certainly was inhibitory to particularly Staph aureus. Which led us then to the final concept of this part, and that was I wanted to recognize another major issue in wound care, and that is that gauzes never seem to fit the tertiary or three-dimensional architecture or the contour of a wound. And it became very apparent that as we wanted to design a new delivery system, we needed to do so with something that actually would be impactful at the tissue interface, or where the biofilm is in its highest concentration, at the basement membrane. And the reality is you can take a digital camera, you can take not only X and Y, but you can take Z, and you can begin to develop a concept of filling a gauze with a particular kind of material that would have the shape of the exact wound bed architecture. We then happen to align ourselves with people who are very strong in ink-based 3D printing. We came up with our smartest, which is now a level 3 and level 4 investigation, and we are now able to, with digital imaging and with probiotics inserted into the reconstituted protein-carrying material, carry now a microbe favorable environment to a wound bed that is a smart and not antibiotic-inducing combination of products. So that's one part of where I've been with looking at beneficial microbes, particularly in my interest. The other is, I must admit, as I'm growing older, I'm looking at what about these newer diseases, or what about these diseases would have to do with cognition. And in the slide in front of you, you'll see that I began to think about the microbial clock in parallels or in combinations. And you'll see that in the upper left-hand circle, it talks about dementia at an older age. And it talks about, in the upper right-hand circle, autism in a younger age. And then in the lower circle altogether, some postulates have said that we could live to 142 in the future. And I kept thinking about, with my microbial clock, what would be the composition of our microbiota at 142, both bacteria and fungi? So I again wanted to think about what I might do in looking at these things, so I went ahead and began to look at what is it that is associated with, particularly in this case, autism associating with a disease of learning. And I had to back up because I began to think about how do we address our microbiota and where does it come from. And one of the very best articles I ever read said, you know what? Mothers delivers twice. In a vaginal delivery, moms deliver not only the infant, but they deliver the first contact with the human microbiota. And how important is that in the development later on of learning and cognition? And how does that relate later on to the lack of cognition in those who are beginning to have dementia? And, although not the topic of this presentation, where do antibiotics fit in? And who and what is the benefit of a cesarean section, if any, versus a vaginal delivery, which in fact delays then the proposition of microbiota being established and the dual hypothesis. When I began to look at then theories of aging, without going into something that I'm just myself beginning to become enormously engrossed in, the idea was that there is the host and there is the environment, and that the host had, as it says on the lower left-hand side, either programmed death or damage-based death that was interactive with the environment, as shown in number 2. What you need to recognize now is that the NIH has said or postulated since 15 that perhaps aging is associated with microbes, and it in fact should be called a disease. And when one begins to think about that and looks at aging, one of the reinforcement of our genetic strength of the host has been the slow acquisition of environmental gene pools coming from our other sources, whereas the acquisition of beneficial microbes being inserted into the lifestyle of the human at some time would be a rapid means of providing a genetic strength and a library which we hadn't thought about. And so this idea is to point out that where we've been is 1 and 2, where we possibly might be going in either looking at something like autism or looking at something like dementia and Alzheimer's disease, we might be looking at the inclusion of beneficial microbes. Along that same line, there is this issue of longevity, and the reality is here that we clearly think that there is, when one kind of separates just the point of our capability of thought, but begins to look at aging as an overall, there is clearly, if you look on the lower right-hand side, some data which is beginning to show that there is a shrinkage of our microbiota. There is a potential acquisition of genes which help some live longer, which is part of that environment, and there is that fraction of the population which clearly has that capacity. But it's the gut microbiota and those as a means of linking through one of the 4 possible pathways associated with long life, which we ascribe at the moment to unique food. But is there some benefit there too? I told you early on that I was at the dental school at West Virginia University, in addition to being in the Department of Pathology, and it became apparent as we began to look at the kinds of oral health that occur in particularly patients who have very poor oral hygiene, and we looked at the composition of that oral health and its impact over time, could we begin to describe a profile or signature of a microbiota which would enhance either detection of an unusual scenario, or would we begin to find some things that were unusual? And as shown here, the first 3 bars are, first 2 bars essentially to the left are those that are from normal patients, and as we move from left to right, we reach to dementia. And I want to point out to you that if one looks at the listed phyla as the upper right-hand side, or the listed family members, you can begin to describe a very real difference in that, in fact, not only is the gut microbiota associated with potential changes of lifestyle, but certainly as a marker, those microbes which are associated with oral health or oral disease are also potentially a marker and a potential target for downfield use of beneficial microbes. So without, again, going too long and getting bound up in this scenario, I want you to realize that we now know that in the neurologic sense and in the gut's interface through at least 1 or 2 pathways, there are those that are listed in front of you for common Alzheimer's disease or, in fact, autism disease. And you can read those at your leisure, but the reality is a bidirectional neurohumoral communication system is involved in both. There is a neurodevelopmental deficit associated perhaps with gut microbes, and that there is an appearance of dysbiosis, both in early age and in the adult age. And the mechanisms by which these might enact, I've shown you on the lower right-hand side, which is the yin-yang hypothesis. So this is where we are, and it comes back to Martin Blaser, who in essence said that we lose our microbiota over time, and that this vertical acquisition as we lose items in a stepwise fashion could be arbitrarily benefited if we had a potential horizontal acquisition. And the key is, if we talk for more and more time, you'll begin to see that we look at our clock and we look at those diseases which are new in life or old in life. There seems to be a pattern of microbes that are missing, and we might be able to associate some disease profiling that would allow us later on to bring into effect beneficial microbes which could complement what might be missing as we grow older in diseases that we have now begin to ascribe as associated with a combination of both the bacteria and the human environment. This slide is something that I put together that being a clinical microbiologist, we so often deal with antibiotics. But I want to point out that as the literature is beginning to become highlighting these problems, please recognize that the microbial clock helps synthesize newborn and clinical childhood diseases, asthma and autism, particularly, are untoward consequences, and in adolescents there are continuing studies that say that all of these may be associated with an unbalanced microbiota, but the bottom line is the feature of that unbalance is associated with overuse of antimicrobials. So in this last section, having given you a little bit, I hope, of background of what I have done and where I am, I want you to realize that we have then tried to explore the educational component, because the information is almost bewildering. There's so much out there that both the medical community, the professional community, and the public needs to continue be appraised of. We've done what most educators do, which is try to put it together in a useful format. And I'm going to talk about then some of the types of things that we've done, but I want you to realize I love this. The picture is a Rubik's Cube, and this is, I think, in the use of therapeutic bacteria, we're in sort of the Rubik's Cube era, where we have gotten a lot of information, but we surely need to be getting more information. Oh, excuse me. So there is a plethora, I love that word, of published articles that address the concept that there may be a place for modulatory capacities of microbes beneficial to diseases, whether they be endogenous or infectious. And if one takes these and begins to assimilate, there is a recognition that in all of this, there is a huge worldwide use of probiotics. Now, probiotics can be classified as one type of beneficial microbe, but what I want to show in this slide is that there are regions of the world that have much more access to probiotics, like antibiotics, and that these regions have become now centers of studies which the United States is beginning to get involved with, but it's striking to me that 30% of the population now uses probiotics. And it's estimated that in the year 2020, the business of probiotics from an industrial point will be about $85.7 billion, which is quite remarkable. What I want you to think about, though, is that as we talk about the microbiota and we talk about us, I want you to think about the organ systems, and the reality is I want you to think about the microbiota as an organ system, unstructured, but clearly it has the capacity and functional needs of Homo sapiens, of all the other systems that we've talked about. And as I mentioned early on, we don't want to trash the microbiota. We want to have stewardship of our what used to be called normal flora, today we'll call our microbiota and our mycobiota. You can think then, if you're talking about organ transplants or restorative microbiology as a form of addressing beneficial microbes, you can think about those that are mini transplants, when we've been working with using means of gums, lozenges, milks, and cheeses, which have an organism called Lactobacillus retroprodentis, which clearly addresses and is as good in some studies as antimicrobials in reversing periodontal disease. On the other hand, if you think about Clostridium difficile and the incredible problems that it has caused in the U.S., we now know that in the world, we now know that this is not a disease of microbes, Clostridium difficile in and of itself. It's a disease of lost phyla. And there are at least 3 to 5 phyla that are incredibly important in balancing what has been a normal flora, or C. diff. All of these produce butyrate, which inhibits C. difficile. So the reality is, as we unmask our diseases, we begin to find that as the population of microbes decreases, the benefit of having a beneficial or probiotic replacement, either from a family or from a constituted laboratory-defined constituency, we could address both mini-transplant and maxi-transplant as a mean of using preventative or beneficial microbes. Having said that, when I began to look at the literature with my students in the dental school and began to find out how many people had very little information and did not know much about probiotics, we began to review the literature about 5 years ago. And again, because of time, I want to point out, though, what we put together as addressed on the website here was the concept that we could look at both a setup where we could make a decision tree that would be separated by professional and public, and we could look at, as you'll see in a moment, in the review of 310 articles as outlined in the tree, we could look at the conditions described in those articles, we could look at the probiotics, the microbes, what were the abstracts, and then also give a value to how good was the study or write-up or summary, and who was doing this, so that people would have the ability to be able to actually search to gain more information on a date and on a website, which would be educational for any group, both professional and those of the public. And we found that there were 11 diseases of which there were significant studies done in the last 10 years on beneficial microbes or probiotics. The largest studies were done in oral, as you would imagine, and more recently in gut. As I already described for you, however, in addition to this, which is available on the website, in addition to looking at what microbes were most useful and what were the studies and how valuable were they in our estimation, most recently our lab and yours truly has looked at wounds and aging, as you now know. And so the bottom line is, I put together also another source within our website of looking at the literature that has described, both in the wound avenue and in the aging avenue, 2 populations. What's out there now, and you can see articles that are listed. You can see what kinds of microbes they used and who they were thought. That is the strength of the studies. But let me point out to you that in either looking at the theories or looking at the published articles on treatment with probiotics in wound care, the reality is most of these are of an international nature. They are limited in scope, and that is the beginning of now an era where we would like to see some of these studies go from industry to medical studies, where we actually have double-blind studied control groups, which takes money, funding, and time. It is a beginning, but we surely have places to go in both the wound theory, getting new information, and the microbes that might be beneficial. In looking at aging in exactly the same point of reference, here are the articles, here are the conditions that were addressed, and under the study, what kind of them were they? Were they reviews? Were they animal studies? And what I would point out to you here is we look at the theories on aging and the published data about probiotics helping, and the actual research. You'll see under strength, or in the middle of these tables, which you can clearly have access to on the website. The bottom line is there is an enormous amount of room for better, newer, and more detailed studies in how aging and probiotics may be a benefit to both the bacteria and to the host. But there is much room for improvement, but I'm still of the opinion, look at the microbial clock, looking at who we are in the hologenetic theory, that we are a dual citizenship. We clearly need to outline that basic cooperation or symbiosis, but we also need to recognize that other investigators are looking at other parts of who and what microbes are also involved in these diseases. Which led me now to the near the conclusion of my presentation, and again coming to the clock. I thought, well, if I've looked at diseases under A, and we've looked at bacteria present, and we looked at bacteria C in the lower left, bacteria missing, and now the increasing evidence of fungi and where they fit, would we be able to put together a unique profile or pattern that would be involved with those four criteria, A, B, C, and D, and be able to define a phylotype for each patient who might have a underlying disease process, that now we have some means of recognizing what phyla are present, what phyla are missing, and could we look at these four parts within that. Which leads me to where we are going. These last two slides are incredibly important because the cancer success in the use of chemotherapy in cancer has led to a population that recognizes the patient needs to be characterized before we use treatment. Not everybody is the same. And in that we now have a word called precision or personalized medicine, and it is carrying over to the non-oncologic or cancer environment, and so that there are definitely ways in which we can address this dual hypothesis, which when subset for each patient will have parts that will allow us to really begin to tailor for every patient a potential beneficial microbe profile, and it's got four parts. And without again the time being now critical, I would invite you to look at this reference. It's positioned here on the bottom. Again, it recognizing that precision diagnosis may be able to help us establish a profile of a prevention. We may be able to better be disease diagnosis in the upper right. We may be able to choose our therapeutics and personalized drug responses, and we may be able to enhance the nutritional benefit of, again, precision medicine, now going into the era of non-oncology, and it would be very much a part of where I would hope we would take this whole concept of the hologenetic theory, which then leads us to the whole idea of artificial intelligence, and that is a whole nother era. But if you can begin to combine the massive science that is in Watson, the massive science that's in Babylon, which is a United Kingdom system, and begin to define it with systems like I've listed in the upper right-hand corner, Gideon, which allows one to make diagnoses from clinical presentation, to begin to match it with antibiotics and the particular etiology or the microbiology, and then our future would be, could we match that to the particular, again, personalized profile of what microbes might be able to be best provided to that patient if, in fact, that's the disease management that would best be part of it. So, in conclusion, really, I hope I've been able to convey to you, well, number one, something I tell all of my students: we live in a microbial world. That microbial world has gained credence and credibility because of the hologenetic theory. The concept is, as you particularly move forward in this, is we don't want to trash our microbiota and our microbiota. We are who we are based on the recognition that we are a human genome that has with it an overabundance and particularly overwhelming microbiota and mycobiota, and that the benefits of sharing both to a benefit to each is what we need to maintain. I love this, unlearning the learned. You now know that beneficial microbes are where we will need to be in the future of medicine. There are those that are in presence that will be markers for good health. Those that are missing that will be markers for potential intervention with a probiotic or something of nature similar to that. the concept that biofilms and microbes that have learned to live as a community and been successful needs to be very, very recognized. And surely, as part of a biofilm, we need to recognize fungi that too can grow in a community of survival, which addresses features that will allow probiotics and biofilms to persist longer. I think we're looking at a whole new concept in wound care management, and it addresses the idea that cells can be encouraged to grow in concert with microbes, which are a part of their cellular environment, that shared benefit. And then lastly, I hope that you'll begin to think of the generation of microbes that are part of our dual citizenship from birth to death. They are who we are. We need to change how we feel about the microbiota. We need to enhance and we need to utilize the genetic strength of that microbial pool. I've listed some resources here that you can use that will be of benefit to you. Not only does the United States have some probiotic guidelines, but the Canadians have some wonderful probiotic guidelines. And in my websites, I encourage you, have you the chance to go to this website, because we particularly highlighted the concept of the hologenomic approach, the co-evolution, and I hope it'll be something that you will address as time is on your side. There are finally three very good textbooks which form a lot of what I have sent to you today. Surely Dr. Blazer has so beautifully addressed missing microbes and what it means. Dr. Perlmutter has addressed how microbes can be particularly addressed and beneficial in the brain. And then two of the previous presidents, the American Society of Microbiology, began to look at a broader sense, but the concept as we age particularly, it's got people, animals, and the environment. And in my slides, remember I had 1, 2, and 3. And that environment is co-involved with the micro and microbiota. We must learn to get along and be part of it and don't trash it. So with that, I think I'm more than honored to have had the opportunity to talk to you, and I hope you'll keep in touch with us. Have a great day. So long.
Thank you, Dr. Thomas, for that informative presentation. It's time for Q&A. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. If we are unable to get to your question, we will follow up with you via email after the presentation. Let's get started. Our first question is, are there precautions and or safety issues in using probiotics as therapeutic bacteria?
And when we spend quite a bit of time thinking about. But the reality is that probiotics and beneficial microbes have been found to be universally very safe, except for a select population. They have been found to be safe in particularly adults and children, but that there are those that have gastrointestinal alterations, cancer patients with the GI tract focus, are those that perhaps probiotics should not be administered and are always considered to be a concern should that be the only alternative. The other issue about probiotics, though, in caution and safety is that storage issues should always be a handle that's necessary to look at when one picks out a particular probiotic, one's going to use them. And then there is, though, the recognition that probiotics certainly have less consequences than the use and overuse of antibiotics, and I sort of draw upon that to show nobody asked me about the safety of antibiotics as much as they asked me about the safety of probiotics. They are, without a doubt, a feature that is beneficial. Thank you. Thank you.
Thank you. The next question is, given their growing popularity and global demand, what is the future of probiotics in medicine and dentistry, given the potential interface with FDA and other regulatory bodies, national and international?
That's another really good question, and it's a tough one. The guiding principles that are still being referred to over and over and over again are those that were put out by the World Health Organization in 2002, which basically said that a probiotic, merely being alive and has benefits that are of value to the person or people receiving them. And that's about it. It's pretty broad. And so most of the use of probiotics has been driven by industry. Obviously, one does not have to go very far to find advertisements about the benefits of probiotics. But there is a limit to what industry at this moment has provided, and there clearly needs to be an interface with medicine and some real clinical studies which would help us get a better handle on what population fits what particular group of probiotics. I think this is where this personalized medicine comes in, where we really now can clearly understand that at certain times certain medications are very much altered by the gastrointestinal probiotic or the gastrointestinal microbiota. And we need to be addressing a use of probiotics in a defined patient target population. I anticipate that the FDA, NIH-driven, and the globe will begin to look at more serious restrictions on who and what is classified as a probiotic. One of the issues that I've had a lot of talk with my colleagues about is that when you do that, you increase the cost. And right now, obviously, that is not a big issue, but should regulatory bodies be involved, that could be an issue. And clearly there are differences within countries. One only has to look at the difference between us and Mexico in the use of antibiotics. So the international theater, given that there are more probiotics and antibiotics used outside of the U.S., needs to be addressed as well. So this is an issue which needs to be a part of a target, but my biggest issue is we need to recognize that we don't want to destroy the benefit by trying to say that one type fits all. That is not the case. We need to be selective, and that's why we have this new idea of personalized medicine, and it will take some time before all of the regulatory agencies begin to come into a flow.
Thank you. The next question is, given the importance of health and beneficial associated microorganisms, what is the impact on the clinical microbiology laboratory whose primary goal, historically, has been to identify pathogenic microbes?
This is a really personal question. I have been a clinical microbiologist since 1969 and been involved with laboratories who have dealt with the evolution of newer test methodology, trying to come up with a concept that would give us better information faster, because unlike our colleagues in chemistry, where they would be able to turn around results within a particular time frame of minutes, the classic microbiology laboratory using culture-defined methods was taking days, and it was something that I heard over and over again. We've now evolved into use of mass spec, the MALDI-TOF approach, where we can recover microbes and then identify them within hours. But we are still left with the concept that the laboratory that is organized has been driven by Louis Pastuer, one bug, one disease, or Robert Koch as in a pathogen. And we're going to have to find a way of probably enhancing or developing a section in the clinical lab which will deal with the health benefits and what is the hologenomic makeup of the microbiota and mycobiota of a particular site or a particular patient with particular disease. We're going to have to go to non-culture techniques beyond mass spec, which will probably be adaptations of PCR methodology. but we're going to have to expand in a way that up to now has not been a part of diagnostic and clinical microbiology. We've only looked for the pathogen. Clearly, with the evolution of the Hologneomic Theory and the concept of personalized medicine, then the clinical microlab is going to have to set aside at least a section which would be devoid of the classic method, but would be devoted to addressing the major issues of what is present and what might be absent in the entirety of the microbiome that is present in the sample provided. That's going to be quite an issue, because the other part of that is, how do you report this? We've looked at new laboratory report forms, and when you think about it, a laboratory report form that is classic and lists a single or maybe two pathogens out of a blood culture, now we'll be looking at a stool and could potentially list, what, 500 microbes? But if we put them into phyla and address the issue that I mentioned early on and begin to look at ourselves as who we are by the phyla and the families that are present, we can begin to get a cluster assessment of who we are and what the patient is. It will be a very different part of what microbiology is, but it surely will have to be done in the not too distant future. And that's one of the reasons I'm still around. I want to see this happen.
Thank you. We'll wrap up with one last question, and that question is, as the emergence of precision and individualized medicine encompasses non-oncological diseases, how will artificial intelligence change the practice of medicine?
That is a huge question, and I got to tell you that some of the most recent articles that I've read have said that the use of artificial intelligence with systems that are evolving are going to be a healthcare strategy revolution. Not an evolution, but a revolution. On the other hand, I've been hearing about artificial intelligence for quite a while. I really got very interested in the concept of focusing on computer-assisted diagnosis in fungal infections, in the early 2000 era, and we were able to develop a scheme where we could really be quite proactive in assessing a particular result and what it meant in a blood culture if a fungal isolate was recovered. The concept here you must understand is that there is no perfect diagnosis, and the literature is full of comparisons between a physician and a computer, and surely the new computers are improving, and our physicians hopefully are improving. The bottom line is there's no question, though, that an enormous amount of money is spent and there are lives lost with misdiagnosis. Will artificial intelligence and the integration of a variety of schemes focused again on personalized or precision medicine be the ultimate answer? I think that's yet to be proven. I'd like to think so. I'm very much a positive person, but I think that the proof is in the pudding, if you will. It has all the intangibles to give us tools that will be helpful, but we've got to put into it a better bit of information. You know, we use the word in microbiology, junk in, junk out. So it's going to take some time in my mind, but the tools are available, and I clearly think that it will begin to have a structured impact on medicine, particularly the clinical laboratory and particularly the infectious disease laboratory. But I'm cautiously optimistic. I want to wait and see if we can improve what we've done up to this time. We need to do that, but I need to see the tools develop that will help us do that.
I would like to once again thank Dr. Thomas for his presentation. Do you have any final comments?
I hope people will become more involved in the changing world of who we are, and will become proactive in finding out the benefits of our microbes. They are our dual citizenships, and they have passports that have allowed them to be with us, and I hope they will really continue to evolve in a sense of positivity, and don't trash your microbiota. Thank you.
Thank you once again, Dr. Thomas. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 14th, 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.